I think I will do my presentation in English, so it will be accessible to as much people as possible. My name is Anwar Mazet. Okay. Well, I have done a bachelor's degree in molecular and cellular biology in Mehmet University in Morocco. And after that, I have done a master's degree in bioinformatics in the same university, and another master's degree in University of Paris in platform engineering in biology, in a PhD in analytical chemistry, bioinformatics, and molecular biology in Tasmania. But today I will talk with you about a part of my PhD subjects, evaluation of the environmental phase and impacts of biopesticides using an innovative approach coupling high-throughput methods, and then about a part of my postdoc. Okay, well, this presentation will be divided into different sections. First of all, a brief introduction, and then chapter 1, study of the dissipation time of biopesticides, chapter 2, study of the impact of the biopesticides on biodiversity, and I will end up by conclusions and perspectives. But I think I got some inspiration from last week's presentation, so I want to start with a little bit of art as well. So this is a painting by Daniel de Magdala in 1847, and it's called An Irish Peasant's Family Discovering the Blight of Their Store. And this period in Ireland history is known as the Great Famine or Potato Famine, which is a mass starvation and disease caused by potato crop failure, which is the main source of nutrients back then, due to potato blight caused by a fungi named Phytophthora ancestor. And this catastrophe leads to over 1 million people dead and over 2 million people who immigrate from the country. Unfortunately, currently we avoid such disasters because of pesticides. Well, pesticides are substances that are used to control pests, and they include a lot of types. We have herbicides against unwanted weeds, insecticides against insects, bactericides against bacteria, etc. Most of these pesticides are used in agriculture, but they can also be used in other fields, such as in forestry, in roads, in railways. Well, pesticides are very important in our daily life to protect crops from pests, but on the other hand, they have some negative impacts, either on microorganisms, where they negatively impact the microbial enzymatic activities, or beneficial insects like pollinators, like bees, for example, and other species like birds and human beings. So because of the negative impacts of pesticides that are not only known only for the scientists, but also for the large population, an alternative to chemical pesticides need to be found. One of the alternatives that we can use are biopesticides. They are complex substances derived from natural sources such as plants and microorganisms. They have higher biochemical complexity compared to chemical pesticides. However, they are supposed to be eco-friendly and less harmful to the environment. Also, they are supposed to be specific in their mode of action compared to chemical pesticides. But we still need to study their degradation and their impacts on biodiversity to ensure that the biopesticides are safe solutions for the environment and biodiversity. The parameter that we use to study the degradation of the chemical pesticides is the half-life parameter that tells us at which moment we will have degradation of 50% of the initial quantity of the chemical pesticides. As you can see here in this table, the different chemical pesticides have different half-life values. However, this parameter cannot be applied for complex substances such as biopesticides, and also this parameter doesn't give us information regarding the degradation product or the impact on biodiversity. So how can we study the degradation of complex substances such as biopesticides as well as their impacts on biodiversity? In Creo laboratory, a new method has been developed to evaluate the environmental fate and impacts of biopesticides, which is environmental metabolic footprinting. This method is based on the use of the biopesticides on a matrix of soil, sediment, or plant. These biopesticides will be degraded either by abiotic degradation that can be photolysis or hydrolysis, or by biotic degradation caused by the microbiome living in this environment, which give us the degradation products. And these degradation products form with the biopesticide compounds, they together form the xenometabolome. And this xenometabolome in its turn form with the microbial metabolism or the endometabolome, they together form the metametabolome. And this metametabolome will be studied by acquiring a profile in liquid chromatography coupled with mass spectrometry. And if we do a kinetic study over time, we can observe the changes in the endometabolome and the degradation of the biopesticides and also the degradation products. And we can determine several parameters. For example, we can determine the resilience time. which correspond to the necessary time for the dissipation of the biopesticide compounds, the degradation products, and its effect on the matrix. In this example here, before the beginning of the experiment, we have exactly the same composition of the microbial metabolites between control samples and treated samples. At the first day of the experiment, we add the organic contaminants compounds that will change the composition of the microbial metabolites and will give us also the degradation products with the time progression. And here also we have changes in the microbial metabolites due to the time evolution. So here we still have 20 days after the treatment, we still have the biopesticide compounds and degradation products. And 4 days, we still have the impact of the biopesticide compounds and the degradation products on the microbial metabolites. But 60 days after the treatment, we return to the same status as the control samples. We have the same microbial metabolites, which means that the resilience time is between 40 days and 60 days. We can study another parameter, which is the dissipation time. It corresponds to the necessary time for the dissipation of the biopesticide compounds only. In the same example, 40 days after the treatment, we have completely disappearance of the biopesticide compounds, which signify that the dissipation time is between 20 days and 40 days. Well, the objectives of this part of my PhD were to study the dissipation time of the biopesticide compounds using modeling approach, to study the impact of the biopesticide on organisms, and to develop bioinformatics, to do bioinformatics development to better study and analyze the metametabolome. So we have conducted an experiment using soil in Perpignan University. We took soil that have never been contaminated by any pesticides, and we had microcosm or 40 grams of moisture soil for each sample. And then we applied a bioherbicide called Beluca. It contained as active substance the pelargonic acid plus formalating agent. The pelargonic acid is extracted from leaves of pelargonium plants. So we applied this bioherbicide to the soil and then taken samples of the soil at different time intervals during a period of 57 days. And for each time step, we had control samples and treated samples, and we carried out metabolomics analysis using specific extraction protocol based on the use of 2 extraction steps, and for each step we use mixture of solvents to extract as much as information as we can extract from the metabolome. Well, the first step is to study the resilience time, which is again it's the necessary time for the dissipation of the biopesticide compounds, the degradation products, and the effects on the matrix. So we need to take into account the whole metabolome. So we have done principal components analysis, and we can highlight 3 different things. First thing is that we have the effects of the time, because we can observe that control samples are separated into 2 different clusters. We have first cluster that contains the early time step from first to 40 days after the treatment, and second cluster that contains the late time step. We have also the effects of the treatment, because we can observe that control samples are well separated from treated samples. And the third thing is that 57 days after the treatment, we still have differences between control samples and treated samples, which signify that the resilience time is not reached 57 days after the treatment. So this is regarding the resilience time. For the dissipation time, which is the necessary time of the dissipation of only the biopesticide compounds, and that signifies that we need to take into account only the biopesticide compounds, and then we need to model their degradation to study their dissipation of each compound of the biopesticide mixture. So, but first of all, how can we select the biopesticide compounds from the initial data that contain whole metabolome? Well, to visualize whole metabolome, I presented the results in a heat map. So the heat map results could be read in this way. So here we have the metabolome, all the compounds that we extract. The green line corresponds to control samples from the first time to the 57 days after the treatment, and the red line corresponds to treated samples. So, and here the blue points correspond to the presence of the metabolites in the samples, and the white points correspond to the absence of the metabolites in the samples. So we highlight a first category of metabolites that are present. They are blue in both control samples and treated samples. So they are likely to be soil metabolites. We observe another part of the metabolites that are white, which means absent in control samples, and they are present in treated samples, and they are also present in the first time step. So they are likely to be biopesticide compounds. and another category of metabolites that are absent in control samples, and they are present in treated samples, but they are absent in the first time step. So they are likely to be degradation products. To study the degradation time, we need to select from this data set only the biopesticide compounds, so we need to select only the green parts. To do that, first we will select the biopesticide compounds and degradation products from the metabolite, based on an output of pre-treatment step that's given the occurrence of each metabolite and control samples and treated samples. For example, these metabolites here, that exist 50 times and 39 times in the control sample, so they are likely to be soil metabolites. And another compounds that exist 0 times in control samples, they are likely to be xenometabolites. And using this method, we have selected 2,858 features as xenometabolites. And once the xenometabolites are selected, we calculated the mean of their intensity in treated samples, and we selected the biopesticide compounds as all the features that have the highest intensity in the first time step compared to all the other time steps. And using this method, we have selected 1,715 features as biopesticide compounds. And for the degradation products, they are simply all the compounds that are not selected by the biopesticide compound selection algorithm. And once the biopesticide compounds have been selected, we calculated their degradation profile with the time progression. And we have done clusterization step to determine all the compounds that have similar degradation over the time. And we generated 7 clusters that present the degradation, basically the degradation products of the biopesticide compounds. And as you can see here, it corresponds to exponential degradation. This can be modeled using a generic exponential degradation function. So we have worked with Swiss Institute of Bioinformatics, and so we consider 5 cases. The first case is we have stable intensity over the time, which means that the biopesticide doesn't degrade with the time progression. The second and the third cases are mono-exponential degradation controlled by one parameter, and thus there is one speed of degradation. And the fourth and the fifth cases are big exponential degradation controlled by 2 parameters, and thus by 2 speeds of degradation. So we applied these 5 cases on each of the biopesticide compounds, and then we have used archaic information criterion parameter to determine the quality of the model. So we applied the 5 cases, and then using this technique, we have selected the best model that well presents the degradation of the biopesticide compound, each feature of the biopesticide compounds. For example, if 2 models have the same likelihood, have the same quality, the model with lowest number of parameters will be preferred and will be selected. So using this technique and the 5 cases, we have for each metabolite of the biopesticide compounds, its dissipation and the quality of the model. And now we have determined the dissipation of each of the biopesticide compounds. We can give it any definition we want. If you want to relate our definition to the half-life parameter, we found that 100% of features reach their half-life before 90 days after the treatment. And if we want to be more strict regarding the dissipation time, and say, for example, it's 99% of its degradation, we found that it's equal to 125 days after the treatment, which means that 100% of features reach their 99% of their degradation before 125 days after the treatment. So in this chapter, we have established a bioinformatic workflow to separate the xenometabolome and the endometabolome and to study the resilience time. And we have established a new parameter to study the phase of complex substances in the soil. And we found that the biopesticide compounds in Beluga persist in the soil for 125 days. So this is the last time. The question is, do they have an impact on biodiversity? So in parallel with the metabolomic approach, we have done metabarcoding analysis using 16S and 18S genes to study the impact on the bacteria and the operative species, and taking only 6 time steps. So the first thing, beta diversity shows that the bacteria are grouped by the time steps and not by the conditions controlled and treated, which signifies that the impact of the time evolution is more important than the impact of Beluga treatment. But if we do that pairwise analysis and we compare at each time step control and treated samples, we found that we have significant difference at the first time step, 2 days after the treatment and 40 days after the treatment. So if we take a moment for the metabolomics data and metabarcoding data, for both data sets, we have the effects of the time, we have the effects of the treatment. And in order to study the impact of the biopesticides and on biodiversity, we need to select only the features that are impacted by the treatment and not by the time evolution. To do that, for metabolomics data, so we have worked with the Institut des Sciences Pharmaceutiques de Suisse Occidentale. The first step is to separate the initial data into the different data sets for each time step. So we generated 6 data sets from the initial data. Then we calculated the variables important of projections, which simply means the variables that are important to separate between the conditions controlled and treated in each time step. And from this data set, so we generated another data set of VIP score where we have all the features and their VIP score, their ability to separate between control and treated samples in each time step. And then from this new data set, if we use it to select the variables, it will be based by the importance of the quality of the variables to separate between control and treated samples at specific time step. So we have added another step of clusterization. So from this data set, we calculated distance matrix. And from this distance matrix, we generated a dendrogram. And then we generated 14 clusters. And we calculated the quality of each cluster and based on the mean and its median value of the VIP score. And we selected only the significance clusters. So the main idea is from the initial data set, we want to select another sub-data set that contains only the variables that are impacted by the treatment and not by the time evolution of the samples. So from this, we generated a sub-data of 1,396 features that contain all the variables that's impacted by the treatment and not by the time evolution. For the metabarcoding data set, the first step is the same. We separated the initial data into the different data set, and then we applied multi-path function that try to maximize, to find the variables that have maximum differences between the modalities, between control and treated samples, with the minimum other variation of the samples of the same modality. And using this technique, we selected 193 bacterial ISV that are impacted by the treatment and not by the time evolution. So using these two sub-data, we have done data integration using mixed omics package. So first of all, we have generated an SPLSTA model, and we determined the optimal number of features, and then we selected the optimal number of the features that needed to be integrated in each of the components for the metabolomics data set and metabarcoding data set. And using these features, we have generated a network correlation of absolute value of 0.75. And as you can see here, this network correlation is dominated by positive correlation and dominated by biopesticide compounds that present 87% and the degradation product 30%. We could annotate some of these metabolites based on their retention time and their mass, and then using another work that's try to characterize these metabolites using MSMS data. So we could annotate some of them, and they belong to polyethylene glycol-based compounds. And currently, we are trying to annotate the other compounds using NZ-MIND and series. And for these bacteria, well, they belong to different genera that contain species that play a positive aspect on the soil or negative aspects based on which species. And this is one of the limitations of metabarcoding because we don't have the details regarding which species are impacted. All we could have is the genera that are impacted. Okay, yeah, so the hypothesis that explain this network is either these bacteria use these chemicals as nutrient sources, or there is chemotaxis interaction between the bacteria and the metabolites, or the identified ISP produce the identified metabolites. So in the second chapter, we established a new method of variable selection to study the impacts of complex substances. We found that bacteria and microeukaryotic species are impacted by biluca treatment, and some of biluca compounds belong to polyethylene glycol-based compounds. And as perspective, so we could apply the developed workflow to study the fate and impacts of another complex mixtures, or we can apply this methodology for another contaminant or pesticide mixture to study their toxic effect. I will back to this point later on. Or to investigate another multi-omics integration tools that are more specified for time series studies. Yeah, or we could integrate another omics tool like metagenomics that can give us more information regarding the impacted species, or metatranscriptomic that give us the impacted functionalities after we look at regions. So as I said, well, cocktail effect is simply if you apply one substance and then you observe its effects on the environment, and you applied another substance and you observe its effects on the environment. The application of both substances on the same samples doesn't mean that their effect is simply the sum of their individual effects, because these substances may interact with each other, which means that their effects can be greater, lesser, or different than the individual effects on the samples. And in the life cycle assessment, which is a method used to evaluate the environmental impact of products from its entire life, from the raw material to its disposal or recycling, we want to determine the impact in the life cycle assessment of substances. What it does is that it takes into account the sum of the environmental impact of the individual substances, which means that it doesn't take into account the interaction between these substances. It doesn't take into account the cocktail effect. And this is very important to take it into consideration, because for a pesticide, for example, if we know that many types of matrices and many types of samples are impacted by more than one pesticide, so it's very important to integrate the cocktail effects in studying the impacts of micropollutants. So the objective of my postdoc is to model the cocktail effects of the organic contaminants in the life cycle assessment. And so, first of all, we have tested a set of substances at different toxicity level on model organisms, the cameroons, where we tested different substances at different concentration, and then we observe the survival probability in 4 days. Yeah, so we have used the different concentration for each substance. And here the case of the profile, when the first day of the experiment, 2 days, 3 days, and 4 days, we observe the number of deaths with different concentrations. Well, the next step is to use mixture of substances and then compare the curves to the curves of using multiple substances to see if we have the cocktail effects, and then from the results of mixture of multiple substances, try to model these cocktail effects in the life cycle assessment framework. So the objective is to first to highlight the cases where we have cocktail effects with the mixture of substances that belong to the same category or just different concentrations. And second thing, to model the cases where we have cocktail effects and to apply the methodology developed to some study cases of quality of water in urbans that are impacted by urban or industry. Okay, so yeah, my current work now is bibliography on the cocktail effects on ecotox and eutox, and bibliography on the different models that can be integrated and are more suitable to study this case of experiments, and to do statistical analysis of monosubstance results. I expect to receive the results in the next few days of the monosubstance results. Yeah, so thank you for your attention.